Welcome back to BioB 160 Principles of Living Systems. I'm Ruth Reitzman, your instructor for the course, and in this episode we'll look at meiosis, the process that eukaryotic cells use to produce gametes for sexual reproduction. The process of meiosis is important in understanding reproduction, and it is essential to understanding genetics and the inheritance of traits. In our last episode, we discussed the cell cycle and mitosis. Many organisms use mitosis for reproduction, which is also called asexual reproduction. Many single-celled organisms, such as bacteria or amoeba, use asexual reproduction for most of their reproduction. In asexual reproduction using mitosis, the progeny cells are identical to the parent cells and are called clones. Sexual reproduction is different from asexual reproduction, since it produces offspring that are not identical to the parents. In sexual reproduction, gametes are produced by meiosis. The gametes contain one-half of the parent's chromosomes. The gametes fuse during fertilization, producing offspring which contain chromosomes from both parents and are genetically different from the parents. The advantage of sexual reproduction is that it results in greater variation than asexual reproduction, and this may give organisms survival advantage. This diagram illustrates a typical sexual life cycle. The body cells or somatic cells of the parents have the diploid number of chromosomes. For the house mouse, Mus musculus, the diploid number is 40. In a sexual life cycle, the testes or ovaries produce gametes, sperm or eggs, through meiosis. The process of meiosis reduces the number of chromosomes to one-half the diploid amount. So for the mouse, the haploid number is 20 chromosomes. During fertilization, the sperm and egg fuse to form a zygote. Since the sperm and egg each contributes 20 chromosomes, the resulting zygote has the diploid number of 40 chromosomes. This diagram shows a cell with a diploid number of 6 chromosomes. The maternal chromosomes are shown in red and the paternal chromosomes are shown in blue. If the chromosomes are lined up by their sizes, we can see that there are 3 pairs of chromosomes, a large pair, a medium-sized pair, and a small pair. These matching pairs are called homologous chromosomes. Humans have a diploid chromosome number of 46, consisting of 23 pairs of homologous chromosomes. Each cell in our body, except for our sperm and eggs, contains 46 chromosomes. Of the 46, one of the chromosome pairs, the X and Y chromosomes, determines an individual's sex. The other 44 chromosomes are called autosomes. We can prepare photos of chromosomes as they appear in metaphase of mitosis to observe chromosome number and determine the individual's sex. This photo of the chromosomes is called a karyotype and is used in prenatal testing to identify the sex of a fetus and to screen for potential chromosomal abnormalities. In this karyotype, we can see a chromosome preparation with the 22 autosomes and the sex chromosomes. What is the sex of this individual? If you look at the bottom of the karyotype, you can see a single X and a single Y chromosome, indicating that this is the karyotype of a male. Now let's look at the process of meiosis and see how it reduces the chromosome number from the diploid number to the haploid number. In our example here, we'll look at a cell with just a very small chromosome number, just 2 chromosomes. So its diploid number is 2. In preparation for meiosis, just like the preparation for mitosis, cells complete a cell cycle with a G1S and G2 phase. During the S phase, all of the chromosomes are replicated, forming chromosomes made of 2 sister chromatids. Just as we saw in mitosis, the chromosomes condense, the nuclear membrane disintegrates, and the chromosomes attach to spindle fibers. However, the difference in meiosis is that homologous chromosomes pair up together and line up on the metaphase plate. In the cells shown here, there's a single homologous pair that lines up. After the first part of meiosis, these homologous pairs have separated, and the number of chromosomes is reduced from 2 chromosomes per cell to 1 chromosome per cell. Meiosis is also different from mitosis because it has 2 divisions. The first division, called meiosis I, reduces the chromosome number from the diploid to the haploid number. Meiosis II separates the chromatids so that the duplicated chromosomes now separate to form single sister chromatids. Now let's look at meiosis in a cell with a diploid number of 6 chromosomes. Here we see the beginning of meiosis in prophase I. The chromosomes condense and the homologous chromosomes line up together. This lining up is called synapsis. This stage is unique to meiosis. While the homologous pairs are tightly synapsed together, the homologous chromosomes, shown in red and blue in this diagram, can cross over and swap chromosome sections. This produces patchwork chromosomes containing a mixture of maternal and paternal characteristics. After prophase I, the homologous chromosomes line up on the metaphase plate and attach to spindle fibers. This is metaphase 1. During anaphase 1, the homologous chromosomes separate and move to the opposite poles of the cell. In telophase 1 and cytokinesis, the nuclear envelope reforms around the chromosomes, and the cleavage furrow pinches in and separates the two cells. Meiosis 1 is called the reductive division because it reduces the chromosome number from the diploid number to the haploid number. If you examine these cells, you can see they started with 6 chromosomes, and at the end of meiosis 1, they each have 3 chromosomes per cell. The second phase of meiosis is called meiosis 2. It's important to note that there's no cell cycle between this stage and meiosis 1, and there's no DNA replication that's occurred between meiosis 1 and meiosis 2. In meiosis 2, the cells formed in meiosis 1 enter prophase 2, the chromosomes condense, and the nuclear envelope disintegrates. The spindle fibers begin to attach. In metaphase 2, the chromosomes align at the metaphase plate, and all of the spindle fibers are attached. In anaphase 2, the sister chromatids separate and move to opposite poles. In meiosis 2, sister chromatids separate, and the end result of meiosis 1 and 2, we've got 4 cells, each containing a haploid number of chromosomes. Remember, we started with 6 chromosomes at the beginning, and now we can see cells consisting of 3 chromosomes, and each chromosome consists of a single chromatid. Now let's compare meiosis and mitosis, and look at some of the events that occur only in meiosis. Remember that the purpose of meiosis is to reduce the chromosome number from the diploid number to the haploid number. The following events occur only in meiosis. First, in prophase 1 of meiosis, the homologous chromosomes pair up and cross over. Second, in metaphase 1 of meiosis, the homologous chromosomes are paired at the metaphase plate. And third, in anaphase of meiosis, the homologous chromosomes separate. These are events that are unique to meiosis, and we don't see these happening in mitosis. Now let's look at a diagram that compares mitosis with meiosis. In mitosis, the chromosomes act independently and homologous chromosomes are not paired up. In meiosis, we can see the pairing of homologous chromosomes. In mitosis, the chromosomes line up separately on the metaphase plate, while in meiosis, the homologous pairs are paired. And finally, the products of mitosis are 2 cells, each with the 2n number of chromosomes. Meiosis produces 4 cells, each containing the haploid or 1n number of chromosomes. What is the diploid number of the cells shown here? What is the haploid number? Count the number of chromosomes present in the cells at the end of mitosis for the diploid number, which is 6 in this example, and count the number present in the cells at the end of meiosis for the haploid number, which is 3 in this case. This chart from chapter 13 also summarizes the differences between mitosis and meiosis. Now let's consider the impact of meiosis on genetic variation in populations of sexually reproducing organisms. We're all familiar with the differences and similarities that we see in our parents, siblings, and other family members. Any child is the product of 2 parents and will possess some traits from one parent and some from another. Another child in the same family will possess a different combination of genetic traits. How does the process of meiosis impact this outcome? And how does this contribute to change in characteristics over time, or species evolution? Some of these questions will be answered in more detail in the next few episodes. Looking at the products of meiosis and the process of fertilization, we can identify 3 ways that these events contribute to organism variability. These are, first, the independent assortment of chromosomes at prophase 1 of meiosis. 2, the crossing over and exchange of genetic information between homologous chromosomes during meiosis. And 3, the random fertilization of the egg with the sperm. Let's look at these in more detail. First, let's look at independent assortment. For an organism with a diploid number of 4, there are 2 homologous pairs of chromosomes. As these 2 pairs line up at the metaphase plate, there are 2 possible orientations that can occur. In possibility 1, both blue chromosomes are on the left side and both red chromosomes are on the right. In possibility 2, the bottom pair has swapped their positions. As we move to the second division of meiosis and examine the cells with the chromosomes aligned at the metaphase plate during metaphase 2, we can see that the products of possibility 1 have both blue chromosomes in one cell and both red chromosomes in the other cell. And in possibility 2, we see that each cell has 1 blue and 1 red chromosome. At the end of mitosis, after both divisions, we can see the 4 possible outcomes. Since these events are totally random, there's a 1 in 4 chance of any one of these combinations occurring. The general formula for determining the possible combinations of chromosomes is 2 to the n power, where n is the number of homologous chromosome pairs. This means that an organism with 6 chromosomes, 3 homologous pairs, will have 2 to the third possible outcomes of chromosomes. And that would be 8 possible combinations. For an organism with 8 chromosomes, there are 2 to the fourth power, or 16 possible combinations. For humans with 46 chromosomes, or 23 pairs, there are 2 to the 23rd, or nearly 8.4 million possible combinations. In addition to independent assortment, crossing over and exchange of genetic information between homologous chromosomes is another factor that contributes to genetic variations that are seen in sexually reproducing populations. The process of crossing over at random locations on the chromosome creates a patchwork, and this blends the maternal and paternal characteristics. If we add to this the random fertilization of a male gamete with any female gamete, each representing any one of 8 million possible combinations in the human situation, we can see that each fertilization event produces a unique and unlikely to be repeated chromosome combination. As your textbook points out, you really are unique. You should now be able to distinguish between the following terms: somatic cells and gametes, autosomes and sex chromosomes, haploid and diploid. You should be able to describe and identify the events that characterize each phase of meiosis. You should be able to describe 3 events that occur during meiosis but do not occur in mitosis. And you should be able to name and explain 3 events that contribute to genetic variation in sexually reproducing organisms. This concludes this episode of BioB 160 Principles of Living Systems. Be sure to watch the BioFlix on meiosis in the Chapter 13 resources of MyLabs Plus. Also read the chapter and do the Chapter 13 MyLabs Plus homework. Thanks, and have a great day.